Hi friends, you're back with me, your friend for biology. So without wasting much time, we are discussing the PYQ. So some questions from the NEET exam. The light reaction, yeah, you got it. It is photosynthesis. The plastoquinone, it facilitates the transfer of electrons from, right? You remember there is a diagram where they are talking about the chemiosmotic, right? So they're talking about the chemiosmotic model for your ATP synthesis. So there they've shown that the plastoquinone, it is facilitating the transfer of electrons between. Okay, let me go here. It is cytochrome B6F to PS1. What I remember is cytochrome B6F to PS1. It is PC, so that is plastocyanin. So this is not the one. PS1 to ATP synthase, no. Then we have PS1 to NADP, no, there is no one here. Like at least it's not plastoquinone. So yes, only one option left here is from PS2 to cytochrome B6F complex. So you remember there is PS2 and going to cytochrome B6F complex, there is this plastoquinone. And you know that when this travels, your H plus actually they are pumped from the stroma into the lumen, and therefore you know that in the lumen the H plus it increases. So as the electrons flow, the concentration of your H plus in the lumen it increases, right? So here you have PS2 to your cytochrome B6F complex. Match the following. You are very good in making matches. We know that matches are made in heaven, but these matches you will have to make. So it is typhoid is a bacteria, Salmonella typhi. Then you have pneumonia, again a bacteria, Haemophilus influenzae. Filariasis, a worm, that is your Vauchereria, and Asclepianthus. And malaria is a protozoa, that is your Plasmodium. Look at the correct options and take the right one. The bilateral symmetry and acoelomates, right? So it is having a bilateral symmetry. Tenophorans are gone because it has radial symmetry. Acoelomate, annelid is gone because it is coelomate. Asclepianthus, remember it is pseudocoelomate. Only one member which is pseudocoelomate is Asclepianthus. Remember that. All before Asclepianthus, your Porifera, Cnidaria, Ctenophora, Platyhelminths, they are acoelomates. All after Asclepianthus, your Annelida, Arthropoda, Mollusca, Chanodermata, Hemichordata, right? They all are coelomates. So bilateral and acoelomate is your Platyhelminths, the flatworms, the liver fluke and fasciola, what you call the tania, the tapeworms, right? The site of formation of glycoproteins and glycolipids in eukaryotes. So modification, right? You attach some carbohydrate groups or lipid group to the protein. So the site where this modification is done, we know that modifications and secretions, that is the work of your Golgi complex discovered by Camillo Golgi, right? So Golgi complex does that. Peroxisomes are having some different enzymes come into microbodies. You have these polysomes, which are many ribosomes attached to mRNA, especially found in prokaryotes. Endoplasmic reticulum, the smooth one involved in lipid synthesis, and the rough one, having ribosomes involved in protein synthesis. So here glycolipids and glycoproteins is your Golgi complex. I'm in a bit hurry. I have a class right now, so stay tuned with me, Professor Kukreja, and we'll be discussing more PYQs. So please subscribe the channel so you have updates about all the coming MCQs. Thank you.